Good afternoon, dear colleagues. Let me introduce to your attention the results of Ukrainian science group on the seed similarity of cultural and natural flora under chloride load in conditions of southern Ukraine. The structure of the presentation includes the following components: introduction, aim, methodology, results, conclusions. Due to increasing anthropogenic pressure on the environment, the climatic conditions on the planet Earth are changing. Saline soils make up almost 25% of the Earth's surface. Unfortunately, the areas tend to expand due to the climate aridization and increase in the average annual temperature of the planet. The south of Ukraine forms a risky agricultural zone which is characterized by a temperate continental climate with hot summers, high solar insolation and significant water deficit. Seeds are an equal product of evolution, the main generative organ of plants, characterized by a number of morphological, physiological and biological features, among which there is adaptation heterogeneity. Therefore, research on the similarity of plant seeds to salt stress is becoming increasingly relevant. Thus, the research aim is to find out the seed similarity of natural and cultural flora under chloride load in southern Ukraine. During the theoretical study of the problem, research and experimental work, general scientific research methods were used. Theoretical analysis and generalization of scientific and methodological literature on morphology, ecology, and the functional peculiarities of plant seeds and periodicals, vegetative and experimental methods, generally accepted physiological methods, quantitative and morphological analysis, mathematical and statistical processing of the obtained data. The choice of the chloride salts concentration is explained by the fact that such a percent is considered stressful for the seeds, taking into account the physiological, biochemical, and anatomical morphological features. The representative selection of cultivated and wild flora was determined by the prevalence of species, taking into account the soils and climatic conditions of southern Ukraine. The obtained experimental data showed that the seeds of cicale cereals sprouted 93% in aqueous solution for 3 days, 89% in 0.1% natri chlor solution, 95% in 0.2% natri chlor solution. The researchers' data presented that the seeds of Triticum durum sprouted well in natri chlor solutions with a concentration of 0.1% and 0.2% by the third day. Seed germination of Avena sativa showed that on the third day the number of sprouted seeds in water was 50%. On the fifth and seventh day it increased to 99%. The obtained results proved that the germination of Hordeum vulgaris seeds began on the third day, with the highest values observed in 0.1% natri chlor solution is 95%. In the aquatic environment, 93%. The faster the germination process occurred with the seeds that germinated in water, the longer the germination occurred in solutions. Our studies showed that during the germination of beta vulgaris seeds, the first sprout appeared on the fifth day in the pure water, up to 30%, in 0.1% natri chlor solution, up to 20%, and the smallest amount was observed in 0.2% natri chlor solution, up to the second percent. Therefore, in beta vulgaris, the best germination occurs in water and in 0.2% natri solution the worst. It is found that the seedling of Phaseolus vulgaris began to appear on the third day in the sample with the water and in 0.1% natri chlor solution. On the fifth and seventh days, in all samples, the number of sprouted seeds increased. According to the results of seed germination of Helianthus annus, it was found that for 3 days the most seeds germinated in aqueous medium. In 0.1% natri chlor solution is 60%, and 0.2% natri chlor solution is more than 20%. On the 5th and 7th days, 100% of the seeds sprouted in all samples. It is proved that the rate germination of Helianthus annus seeds is characterized by inversely proportional dependence on the salt concentration in the medium. Seed germination of Valeriana officinalis was observed for 12 days, but during this period the highest values were obtained in the aqueous environment, the lowest in 0.1% and 0.2% saline solutions. For the anti-germination period of Matricaria recuta, seeds which were 7 days, the highest values were observed in water. The average values were in 0.2% natri chlor solution, and the lowest values were in 0.1% natri chlor solution. The researches of the salt hermesis seeds of natural and cultivated flora in the conditions of southern Ukraine showed that the number of germination days, the environment, and the definition of sodium and chlor ions influence determined the opportunity distribution of the studied plants. It has been proven that the diagnostic signs of florid hermesis were most clearly manifested in representatives of cultivated flora than in natural ones, which is due to the realization of the relationship between the genetic program and phenotypic manifestations that are associated with the special climatic conditions of southern Ukraine, which is of practical importance when developing strategies for cultivation, agricultural and decorative crops on saline soils, and in the future should be taken into account when selecting a plant assortment for the land of southern Ukraine. Thank you for attention.